Hello, this is Mr. McLean, and I'm back to give you a little bit of a review on the material that we covered in section 4 as well as 5 of the Tour of the Cell for AP Bio. So where we last left off in our previous video, we were finishing up and leading into section 4, which starts to talk about the endomembrane system. And I think it's a good example of a collection of different structures that work within the cell to accomplish a variety of tasks which are linked. A lot of times we look at things individually and we study them individually and we don't necessarily see the connectivity. So I think this is a really good example of how we can see structures working sort of from a system standpoint. So you see the list of what we're going to be focusing on here. We got the nuclear envelope, and in the previous video we talked a lot about the structure of that, so I'm not really going to address it too much, but we want to focus in on the different parts of the ER, the Golgi, the lysosomes, vacuoles, vesicles, and then the plasma membrane. And the big thing to keep in mind with this endomembrane system is what we have here at the bottom, and that is everything is connected. So it's either continuous, which means it's going to give you a direct connection, or it's going to have these vesicles. And we're going to get a chance to see a little bit of that as we move through this section. So the first structure that we're going to take a look at, assuming that we've already reviewed the nuclear envelope, we want to move into the ER. And a lot of times when you look at the ER, we should be thinking of it sort of as our membrane factory. It's going to be an area of a lot of biosynthetic metabolic functions. And it's going to have a significant amount of membrane in and of itself. So if you look at it, about 50%, maybe even more, of the total membrane of a eukaryotic cell is going to be contained within that ER. And as I showed you in the previous video, when you look at the structure of the nucleus, you're going to see that the ER is continuous with the nuclear envelope. So there's actual connection points that are going to allow movement of materials and information, if you would. So looking at the ER, even though it's continuous without any breaks in this, you're going to see that there are different distinct regions. So we're going to have our smooth ER and we're going to have our rough ER. And the reason why one appears smooth is it doesn't have the ribosomes on the surface, and the rough is going to be studded with ribosomes on the outside. So those are the same ribosomes that we talked about before as far as attaching. And if you look at this picture right here, it's showing us that we have sort of a series of different transitions that are going on here. So we have the smooth with nothing attached to it. We have the rough, which if we zoom in a little bit, you're going to notice the rough is going to have the conspicuous little ribosomes on the surface. We're also going to have what's referred to as our transitional ER. And one of the big functions of the ER is to be able to take proteins that are manufactured on these ribosomes and to push them into this internal area. And they get modified. So they kind of get pushed through there, and they get modified in a variety of ways. Maybe there's sugars added to the surface, and by the time they are ready to be exported out of the ER, they're going to kind of create these little bubbles. So if you look like how this is doing this right here, if this was a protein inside of here, what these membrane structures are going to eventually do, because they are fluid, is it's going to do one of these types of things. It's going to break off as a transport vesicle. So the transport vesicle is going to then go on to the next layer of modification. So this may be traveling to a Golgi, or it may be going to the end delivery point, depending upon the system. One other thing that I wanted to focus on before we leave this slide, two terms. This space that exists all throughout this inner area right here. So if you think about it, it's almost like a big tunnel system. You've got all this inner space right here. That is referred to as the ER lumen. It's also referred to as a cisternae. So both of those words kind of go interchangeably, but the lumen or cisternae would be the inner area right through here, where you're going to get a lot of that modification and functionality coming in. So let's start off up here with the smooth ER and look at the main things. Big things that you want to keep in mind is we're going to be able to synthesize different molecules. So a lot of lipid synthesis. And when you're thinking about lipids, you're talking about, triglycerides and phospholipids, but you're also talking about steroids. So when you start to think about the smooth ER, one of the things I want you to be able to do is, for each of these organelles, I want you to think about what kind of cells would have high concentrations of these different ERs or these different organelles. And, when you start thinking about the reproductive organs and the ability for them to produce different sex hormones and steroids in some ways, you're going to see that the smooth ER is going to be very prevalent in those cases. They're going to help us break down and metabolize carbohydrates. We also talked extensively about the impact on different toxins as far as detoxifying alcohol products or barbiturates in terms of drug products. What we would do is we would see the addition of hydroxyl groups added to these different molecules, and what that'll do is that'll boost the solubility. So by doing that, we're able to get this out of our body in the form of urine and to help, like you said, flush the system. So you would expect this to be pretty prevalent in the kidney, okay? You're also going to find elevated levels of this, most significantly in the liver. So our liver is going to be really the area we're going to get a lot of that smooth ER. Other things that are going on in the smooth ER is it stores calcium ions. All right, and we're going to find out that part of our homeostatic mechanism is for us to regulate calcium levels, which eventually plays a really significant role in how we do nerve transmission and how we have muscle contractions. So when we need to reduce the free calcium, we'll take it into the lumen of the ER. When we need to elevate it, we'll release it from the lumen of the ER. So it serves as a pretty good storage area. From the rough ER standpoint, what we're going to find is that they're going to mostly be responsible for making the proteins. And you see this word here, glycoproteins. What that's telling us is protein with sugar. Okay? And we go through, we make the proteins, we modify them a little bit, we push them through that tunnel system, and then we distribute them in transport vesicles. So those proteins that are being released off of there most often are either going to be part of the membrane of the cell, or they're going to be secreted. And then, like I said, there are some other scenarios where they stay internally, but most of the time they're going out of the cell or becoming part of the membrane of the cell. So if we go back to this picture right here, we saw that this transport vesicle was being sent, and while it's been modified and changed a little bit, it is not necessarily ready for its final destination. That brings us to the role of the Golgi apparatus. And if you look at the picture of the Golgi, you're going to find, once again, it's going to have these internal areas bound by membranes. They create these little cisternae. And this collection of these membranous areas excuse me, is going to allow us to see the movement of our protein coming in on the cis face. Okay, so the cis is kind of the receiving side. We're going to have things coming in on the receiving side, and they're going to be exiting on the trans side. So from this point on, after it's been modified and changed, we should see a protein that is going to be more appropriate for whatever its next function is going to be. If we go back and we look at what the Golgi apparatus is, big things to keep in mind is it's going to be made up of multiple cisternae, if you want to think of it that way, those membranous little compartments. And we're going to take products of the ER and we're going to make sure that they're modified. They're going to be ready to go. We're going to, in some ways, actually be able to assemble bigger molecules. So you're going to get your macromolecules. And the old analogy that they kind of serve as the post office is definitely applicable, because you're going to see that they're going to package materials and they're going to put them into these transport vesicles so that we can export them out of the cell. So that's the role of our Golgi. Now, I mentioned that most of our protein products are going to be secreted or become membrane structure components if they're released in the process that we followed. But occasionally, they actually remain inside the cell and they serve to help with the digestive functionality. So if they form a structure of a lysosome, what we're going to find is the lysosome is going to be a membranous structure that's going to have hydrolytic enzymes. So those are enzymes that can actually go in and digest the macromolecules. And what they will do is they're going to help us metabolize things that are introduced into the cell, whether it's a foreign particle or a food item, or in some cases they're actually going to help with the cellular cleanup, if you would, of parts that are no longer functional. So if we look at this picture, we can see both things happening. Here we have the process of phagocytosis. So here's our foreign particle, and it is moving through the membrane. So this particular process is a type of endocytosis. So literally we're kind of creating this bubble in the surface of the membrane, and some of that plasma membrane material finishes off this barrier, if you would. The lysosome is going to attach to this particular vacuole, this food vacuole, and the hydrolytic enzymes are going to work to digest it. So we're going to be able to break those parts down, and that's how the cell is able to get nutrients in many ways. Now, the other thing that we can find is a process of autophagy, and what that's doing is the parts inside the cell occasionally need to be replaced. They either go bad, they're damaged, they're no longer functional. So the cell has really no tolerance for inefficiency. So what we'll notice is that the lysosomes will fuse to more or less a vesicle containing worn out parts, and that'll do digestion. Other big thing to keep in mind about the way this works is most of those enzymes work in a low pH environment. Okay, so in the cytosol of the cell, it's close to neutral, so that's not going to be ideal. But when we compartmentalize and we package it up, we can actually have a great environment for digestion. We're going to find out a little bit later when we study some of the genetic disorders. There are lysosomal disorders, which we will revisit and look at more specifically. We also notice that when we start talking about apoptosis, which is programmed cellular death, we're going to see the role of the lysosome showing up in that area. So that'll be a little bit later. And then just kind of wrapping this section up, we take a look at the idea of vacuoles. And vacuoles are storage areas. We have food vacuoles, we have contractile vacuoles, we have central vacuoles, and the idea is that they're going to help us hold nutrients and materials. In a plant cell, you're going to have a much larger central vacuole, and what you notice is it kind of pushes that nucleus off to the side. So the whole idea that the nucleus is always going to be in the center is really not accurate when you're looking at a plant cell. So that will bump it over to the side, and we'll get a structure that looks like that. Aside from ions and other nutrient material, occasionally you're going to get some toxins in there and some substances that might be what we would consider irritants, which might be defensive in some cases for an herbivore or against an herbivore. Okay, so that kind of gives us our rundown of the endomembrane system. I think if we finish off this section just by looking at this slide, what we'll notice is there's a connectivity between all the different structures. We're going to notice that there is a continuous nature between the nuclear membrane and the ER. You clearly have two distinct regions of the ER with totally different functionality. Even though they're connected, they have different functions. You're going to see the ribosomes, which are not part of the endomembrane system, but in this particular case, they are serving a function. We can see that the formation of vesicles will exit the ER, and they're going to be received on the cis side of the Golgi. As they get modified, changed, add new carbohydrates, what have you, they're going to exit the trans side. From there, really there's three things. Either A, they stay inside, they be a lysosome. B, they become part of the plasma membrane. Or C, they exit all the way out. So I think that's an important area of the cell structure. You definitely want to make sure you know all the components of the endomembrane system and how they work together. Thanks.